Good morning, everyone. My name is James Mainprice, and myself and Alison Chung and Amelia are here on behalf of Martin Yaffe, who is the PI for the Image Validation Core. Unfortunately, he couldn't be here today. He had a trip scheduled to go explore north of the Arctic Circle on a icebreaker ship, which was meant to be in August, but the ship broke, and so he's on a different ship. And he's currently somewhere where there's very little internet and enjoying the far north. We're going to talk a little bit about an introduction to the IVC program. And basically, we're largely located at Sunnybrook under the Burrell Lab, which is the Biomarker Imaging Research Lab. So the image validation core is really to provide a connection between medical imaging, so MR, X-ray, and so on, to what we see in pathology or in seeing in terms of outcomes. So in terms of maybe biomarkers that we see in proteomics or genomics and so on. And what we want to know is, why do we see certain cancers in medical imaging, and why do we miss certain cancers in medical imaging? And what is the connection between what we see in those images and perhaps the prognostic factors that we have identified at the sort of the bio side and the pathology. And one of the contexts that we try to do in terms of looking at the process is try to maintain as much as possible the spatial context throughout this imaging validation. So knowing where the cancer is in the medical imaging and knowing where the cancers are in the pathology, and recognizing things like heterogeneity and various variations in tumors and locations and aggressiveness and so on, and trying to make those connections. The idea here is, is well embedded into the OICR program in terms of early detection and understanding how to find these cancers early. Very often, imaging is where we detect the cancers, the first signs of cancer. And it fits very nicely in terms of developing that translation. So having a goal towards moving into the clinic. And the focus is really is to provide several key things that we think that we're relatively skilled at now that we've been doing it for a number of years. One is whole mount histopathology, meaning that we have these large sites for the sectioning, very large lumpectomies or large organ systems. We've done brain and prostate and so on. And then providing both spatial proteomics on the pathology side, and then going back and looking at the images and providing radiomic approaches, or even as we're starting to get into it, more AI, looking at both the medical imaging and using those tools there, as well as looking at the pathology, using those tools to help essentially process a huge amount of information that are in the images. So the connection here really is we provide several services through the IVC, and this is done through either a service, sort of a fee-for-service approach, or through collaborations, and that really depends on how complex your problem is and what you want to achieve with that. And maybe a service, something really simple that needs to be done that's well understood and well contained, to a collaboration where we need to develop something new, maybe a new pipeline to validate that imaging biomarker that you're interested in looking at. I won't go into all of the features there, but we look at biomarker discovery, research pathology, and correlative radiomics. So in terms of capacity, we have a fairly large lab. We're lucky enough to have a space that was, back when it was first developed, a large unoccupied space at Sunnybrook Hospital. Now that space is a premium, we're very lucky that we have that space. Similar to the diagnostic development labs and the genomics lab, we are also certified clinically. So we have IQMH certification, meaning that it's of diagnostic quality and can be used in the clinical program. And we're closely linked, obviously, with the image processing and analysis lab and a genomics core lab that we have on site. I should mention, too, that we all are well embedded into the programs here at OICR, and we certainly interact with the diagnostic development lab and genomics and so on. And we try as much as possible to keep all of this data as best as possible and try to translate into something that can be shared. And that's where we have a clinical research repository, and this is what we refer to as the biomatrix at Sunnybrook. And this is something that we've been working on and trying to develop for a number of years. And in fact, we finally got the hospital, the main hospital, interested in this, and they're looking to translate a lot of the clinical data, information on patients and so on, into that system so that we can make the connection between the research side and the clinical side. We have a fairly large group. This is IVC's expertise that are on site. This is a bit of an older picture, but I think it illustrates the breadth of the group that's working on this. We have physicists and lab technologists, biologists, clinical research coordinators to help us with clinical trials and so on. And I'm going to turn this over to Allison to talk about a little bit more about the equipment and services.
Morning, everyone. So I'm going to show a few slides highlighting some of the equipment over at IVC at the Burrow Lab. For those who have signed up for the virtual tour later, we will also be showing that in the video that we'll have for the tour video. And I also wanted to mention that some of these histopathology services is also available at OICR for the tissue portal at diagnostic development. So as James has mentioned, one of the unique strength or development at Burrow has been the whole man processing technique. And it has been based on the development of this gel that we actually embed huge pieces of tissue instead of the tiny ones, and then cut it using a tissue slicer. And then we were able to preserve the spatial context of the entire specimen. Because it preserves the entire specimen with spatial orientation, this technique has been used widely for imaging validation of imaging techniques because we can make macroscopic comparisons between the whole in vivo image as well as the surgical specimen. We also have built an in-house micro-CT machine so that we can make these correlative studies. Given all the advantages of using whole man technique, it has not really been widely used in the clinic, mainly because of the cost and the complexity of the procedures. But we found that this has been a very useful research technique to be used in the lab for more research-based studies. In order to process all these huge specimens, we have various types of tissue processors. We have these microwave and hybrid tissue processors, as well as the regular ones, and the tissue embedding center as shown here. All of these standardized protocols have been approved by the anatomic pathology at Sunnybrook for both research and clinical diagnostic specimens. With the large blocks, we have been using this macrotome, as you can see over on the top left corner. It's extremely tricky, as you can imagine, to cut these very, very large, thin paraffin sections. And our MLT are very experienced in doing that. We also have a cryotome, cryostack for processing frozen specimens. In terms of immunohistochemistry services, we have a Ventana Discovery Ultra in the lab, which helps for development of IHC protocols. We also have a number of other autostainers for IHC staining and also for HME stainings. We have a number of digitizers in the lab for high throughput slide scannings. We also have a tissue scope, which is specifically for digitizing these extremely large glass slides that we have in the lab. We also offer microscopy training for students and staff who wanted to learn more about scanning. So biomarker multiplexing has been one of the areas that we have focused on for the past 10 years. Currently, we have 2 systems running in the lab. Both are using the cyclic immunofluorescence staining technologies. We have the cell dive from Leica Microsystems and also the PhenoCycler Fusion from Akoya. We have been making some studies in comparing the performances of the 2 systems, and we will continue to try to complement these systems with other multiplexers or with other single cell profiling technologies. I just want to mention also that we are the only lab in Toronto so far that has these two systems in the lab. And the PhenoCycler-Fusion is hopeful that we will be upgrading it to the 2.0 version, which has both the capability to look at RNA, and also we're able to analyze two slides at the same time. We fully provide support to clinical trials and studies, including sample collection, tracking, storage, and so on. And we have a QA system, a QA program supported by a daily, weekly, and monthly QC process, and we also maintain the annual preventative maintenance process. We have a genomics core inside Burl. It's really not part of adaptive oncology, not part of OICR, but we collaborate very frequently with the genomics core. It's run by Dr. Arun Sav, and he has a number of the NGS equipment and also NanoString, and they also recently got the automatic DNA and RNA extraction systems. So now I just want to spend a little more time on talking about the pipeline that we have developed over the years for analyzing multiplex images. So as I was mentioning, we have two systems running in the lab, and we have made some progress in terms of the wet lab procedures. For example, our multiplexing specialist, Dan, she has developed a new improved photo bleaching method, which will soon be published in the journal AIMM. We have also incorporated H&E staining now to all of the slides that we studied using multiplexing. So we have quite a big repository of images of both H&E images and multiplex quantitative analysis of the same slides. In terms of single cell segmentation, just want to point out that we have an algorithm called DeepCSAG, is specifically for segmenting single cells of multiplex images. It's developed using a machine learning approach and is open source, is publicly available for those who are interested in trying it out. Recently, we have spent more time on looking at spatial biology tools. The most recent ones is the graph construction, graph K-neural network, graph K-nearest neighbor analysis of the cell arrangements. And we have also recently improved the UI so that the users can more easily change the parameters when they run the algorithm. And then lastly, I just want to mention that we have been spending currently a lot of time on building tools to look at data integration or multi-omic analysis. We're an imaging lab, and the advantage is that we have also in vivo images of a lot of these cancers. So we have both radiomic, molecular, and also proteomic data sets of the same cohort, and we're trying to find more ways to be able to find a signature for a better characterization of these tumors. And I hope to tell you more about that in the future. I also want to mention that Dr. Ann Mattel, a co-director of Burl, her lab has over the years built a battery of different tools for doing digital pathology analysis. So recently they launched a website called the DP Lab Tools, and it's in the format of Python toolkit for all these tools that they have developed. And if you're interested in using them, please go take a look. For the last part of my talk, I just want to give you some highlights of the projects that we have done at IVC. So with adaptive oncology, with a number of groups at OICR, we have actually started looking at intratumoral breast cancer heterogeneity. And this is done using the whole man process lumpectomies that we have at Burl. With multiplexing, we're able to measure the biomarker expressions from different regions of the same tumor, and we found marked levels of variations for these different biomarkers. We also looked at some other more clinically relevant findings, like we have multiplex measured HER2 protein. We compare it to like B2 gene expression, and we also try to compare that to see what pathologists are scoring those specimens for. In the middle is a panel of some examples that we use multiplexing to phenotype immune cells, and we try to find out where they are localized in the tumor. We also have looked at the cancer-associated fibroblast. We found a group of these CAPS clusters that seems to be more prevalent in different various molecular subtypes of breast cancer. And then lastly, the panel on the right is an example of a multi-omic study that we have, studying samples from a imaging trial called TMIST. We were able to collect radiomic features. We did RNA sequencing, DNA targeted sequencing. We have some research-based subtyping and prognostication scorings. We also have multiplex IF looking at the TME. So we do have a panel, a small sample size that we're looking at integrating all these data types to see if we can find a significant feature out from it. James has also looked at some of these radiomic features and found that there may be some relationship related to the 95 gene recurrence risk score.
Okay, so the last example of sort of the pipeline is looking at spatial pathology and medical imaging correlation. And this is where the whole mount pathology really comes into play, but we've also worked with some of the clinical pathology images, which are the small slides, which are usually meaning that the organ system or the cancer, the lumpectomies are diced up into very small sections, but we can sometimes recapitulate the 3D volume. And just shown here on the, see if I can point a pointer on here. Here we have a series of serial sections from a breast lumpectomy. We've done a lot of work in breast in the past and continue to do so. And we're also working in breast prostate. So these are two AO programs that have been heavily involved with in that. On the top right here, this little movie loop spinning through here is a rendering of these serial sections, where the serial sections were annotated by a pathologist to indicate sections showing here in the 3D. The red is the invasive disease, and then with two very distinct foci. And then there's two smaller sections with in situ disease in green. And then down in the bottom here is a typical layout of an MR view showing the three views, axial, coronal, and sagittal, or rendered in 3D volume rendering with the lesions here segmented by radiologists down here. And you can see there's a very nice correlation visually between the two foci seen here very nicely in both views. So we try to do this as much as possible. Sometimes it doesn't work in terms of getting true sort of geometric 3D relationships, but we often try to get something that Dr. Masoom Haider coined is called the cognitive correlations, knowing that we have this disease here and this disease there and so on. Just like to indicate, moving forward, you have new collaborations, and one of the very newest ones is with investigator Cheng-Shi Chan, who's in the audience, and this is looking at Raman imaging, which really has a lot of potential here. So this is sort of label-free histopathology using the phenomenon of Raman scattering in this, and so this is something that could be very quickly be able to get signal from images from tissue samples that don't have to be processed. You basically put the tissue samples in the microscope, get the Raman scattering signals, and generate images that can be an analog to H&E staining and so on. You can see some of the examples that are very nicely shown here. And then just the last slide is connect with us. As we said, the principal investigator is Martin Yaffe, and he's happy to have a number of discussions. Myself and Allison are here as well. And then if you're interested in any of the services or collaborations, we have the details here. Contact Julia and we can move forward from there. And I think that's it. Oh, and the funding for this. And this is just a number of the connections we have for the various projects, both in the IVC and connected to the IVC. Well, thank you.